Welcome and good morning or good afternoon, depending on where you're tuning in from, and welcome to the Invasive Mussel Collaborative webinar series. Today we have presenters Ashley Elgin with the National Oceanic and Atmospheric Administration, Diane Waller with the U.S. Geological Survey, and Jeff Tyson with the Great Lakes Fishery Commission, who are going to discuss recent tools and accomplishments of the Invasive Mussel Collaborative. My name is Sam Tank, and I am joined by my colleague Patrick Caniff with the Great Lakes Commission. And on behalf of the Invasive Mussel Collaborative, we want to thank you for participating in today's webinar. As a bit of background before we get started, the Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for the management and control of invasive dry-seeded mussels in order to produce measurable and ecological benefits. The Invasive Mussel Collaborative published a strategy to do just this and advance the control and management of zebra and quagga mussels. The strategy was released in 2018. The strategy identified 5 goals and a series of subsequent objectives. To make progress on these goals, the collaborative established 3 work groups: the research work group, the dry-seeded toxicity testing work group, and coastal site priorities work group. The research work group is charged with coordinating existing research and developing targeted research plans and projects that address Great Lakes management priorities. The dry-seeded toxicity testing work group is charged with reviewing how toxicity testing of potential control methods on mussels have been conducted across various labs, discuss strengths and weaknesses of different approaches and methods, and identify and make recommendations where methods could be further refined and standardized, and finally, develop associated guidance for use by researchers. Then the third work group, the coastal site priorities, is charged with reviewing information on where dry-seedids are impacting Great Lakes resources, evaluating sites based on this information and additional criteria, and developing and implementing a system for prioritizing sites for potential management activities. Our speakers today will provide an update on the recent accomplishments of each of these 3 work groups. Today's webinar is part of an invasive mussel communication network facilitated by the collaborative. And the purpose of these webinars is to facilitate learning and information sharing on topics of common interest within the invasive mussel community. The webinar will be recorded and archived along with past webinars on our website. So if you'd like to learn more about the collaborative, or if you're interested in staying in touch with our through our communication network, we encourage you to visit our website and join our listserv. Our website can be found linked at the end of the webinar. So while we always strive to minimize technological issues during these webinars, we do apologize in advance if any may arise. But I just want to cover a few logistical items before we get into our presentations. Audio for this webinar is going to automatically be streamed through your computer speakers unless you choose to call in using the connection information that was sent out in your registration confirmation email. In order to minimize any potential for interruptions and background noise, we've automatically muted all attendees, as well as disabled webcams, and attendees will remain muted with webcams disabled throughout the webinar. We do encourage and will accept all written questions from attendees during the webinar. So if you have a question, even if the presentations are not complete, go ahead and throw it in the chat, and that's signified by the little chat bubble circled in green on the screen right now. And we'll do our best to make sure that we answer them before the end of our webinar, which is 2:30 p.m. Eastern. Even if questions, or even if you have to leave before your question is answered, you can go ahead and review the recording of the webinar to hear your answer. So if you do experience any technical difficulties, you can go ahead and send us a message through the chat box. You can send a direct message to Patrick Caniff and he's able to assist you, or you can send an email to mussels@glc.org. So that ends our introduction of the Mussel Collaborative. Now I just want to turn this over to our first presenter, who is Ashley Elgin. Dr. Elgin is a benthic ecologist who has been studying the impacts of invasive species on native food webs for more than 15 years. As a research scientist at NOAA GLERL, she oversees a long-term monitoring program for benthic invertebrates in the Great Lakes, with a focus on invasive quagga and zebra mussels. She also conducts field and lab experiments to address the many remaining knowledge gaps about these species. Dr. Elgin is based at NOAA's Lake Michigan Field Station in Muskegon, Michigan. Ashley serves as the co-chair of the research work group along with Diane Waller. And go ahead, I see you're sharing your slides. They look great, Ashley. You can go ahead and take it away.
Okay, good. Sam, thank you for that great introduction, not just for the talk today, but for the collaborative in general. And so today I am the first of three of us co-chairs to present recent IMC tools and accomplishments. I'm going to focus on the research group outputs on behalf of myself and my co-chair Diane Waller of USGS. So this I can go through very brief, thanks to Sam's wonderful background information. But briefly, the collaborative was formed and established in 2014 by multiple agencies with this goal to advance scientifically sound technology to control invasive mussels. For more information about the collaborative and a wealth of resources, including some of the products we're presenting today, can be found at the invasivemusselcollaborative.net website that is developed and hosted by the Great Lakes Commission. So the structure of the work group, as Sam already mentioned, we have, there were 3 work groups developed. This came out in 2019. The collaborative decided to implement this approach of using work groups to address areas of identified need. And these groups are for research, toxicity testing, and coastal site priorities. And our ultimate goal is to develop products for collaborative use. So focusing in now on the research work group, our charge is to advance effective Dreissena mussel control methods to restore biodiversity, ecosystem functions, and services. The specific tasks are to compile ZQM, or we use that throughout the presentation to stand for zebra and quagga mussel research efforts. Also to conduct and share the outcomes of research and identify focal areas to prioritize research gaps and needs. Ultimately, in doing this, the desired outcomes in that our research efforts are going to be better aligned across agencies and institutions. We want to make sure that any efforts we have are more synergistic than they are redundant. Also that the stakeholder community is more aware of research outcomes. And what's important about being aware of projects as they start up is then you don't have to wait until results are published to know what initiatives are out there, especially if you're trying to be at the forefront and the state of the science for Dreissena mussel control. And finally, that high research priority questions are identified. So first, before I go on, I must acknowledge the research group members. So here's a list of the members. I won't read through everyone here, but I thank to everyone for your engagement in the process here, your feedback and your guidance along the way. We also receive critical staff support from the Great Lakes Commission from those listed here. So Sam, you've met already, Ceci, Jill, Patrick, and Kate. So thanks again to all of you for being the engine that makes this run. So the first product I'm very excited to present on today is the Dreissena Project Coordination Mapper. There's just a brief history that we've had efforts to try to gather research projects and display them in the past, including before 2020, we had an initial call out for research projects, and also the Invasive Mussel Collaborative website had a project map that was more focused on applied control and response efforts. In 2020, there was the research work group. Now that we were formed as a formal work group, we put out a call for new products because any meaningful product inventory requires very regular maintenance to make sure that ongoing efforts are represented properly and new projects are being captured. From this call, we got information on 74 projects that were from across North America. In 2022, then, we combined the existing project databases that we had in hand, along with the applied control response database, into a new ArcGIS platform that is highly visual and very interactive. And I'm going to take you on a tour of that today. It has an extremely long web address if you go to the formal web address. So luckily, they have a truncated one here that's bit.ly/IMC mapper. You can also navigate to it by going to the homepage of the Mussel Collaborative and go to research projects, research and projects. When that drops down, select the Dracaena Project Coordination Mapper. And one thing is, we go forward, another motivation of this is that this tool, our goal here, it can be used by a researcher to find potential collaborators, or a manager can go in and use this tool and learn more about relevant applied control projects or specific control techniques of interest. So when you first go to the website, you are greeted with this helpful opening page that says, welcome to the mapper, get familiar with the tools and the functionality here. And so you can spend time to review this, or you can X out of it at the top and move on to the mapper itself. Again, I'm including a link to it in the corner or the bottom here if you didn't have a chance to catch that already. So when you get to the mapper that's oriented in this way, in that on the left-hand side, there's an interactive map, and on the right-hand side, there's a project list. Since we were combining projects of two different types, both research and applied control, we took the approach of having two separate lists of projects and in symbology to represent in two colors the two types of projects lists that are represented here. Because each one has its own fields that relevant information that's just specific to that type. So to introduce you to the two types we have, there's the research project that's shown in brown. Those are the brown dots on the map. And then when you're in the project list, you would click on that, the brown tab here that says research projects, and you know you have the right list if all the detail boxes are brown throughout the project list. These are projects where the primary goal is to advance Dracaena mussel control technology, enhancing strategies for managing mussels, and or investigate the biology and ecology of mussels. Whereas an applied control project, that's more of a non-research project that focuses on applied control efforts or rapid response to control remove mussels. One note to help you navigate and use the page is if you're on the map and you want to click on any one of these points, it will bring up and then highlight the project in the list on the right-hand side. However, you'll need to be in the research project tab if you click on a brown dot for the project to appear. And if you want to click on a blue dot, just make sure you've clicked on applied control projects beforehand so that the right project list is up. So I want to feature a function that you can use on the map itself. Let's say you have an address, a specific place or water body that you want to look at on the map to see both research or applied control projects in that area. So in this demo, I put in Lake Erie to that box, I hit enter, and it brought me here. And this list for Lake Erie, you can see both the research projects in brown or the controlled projects in blue. And I selected a project of interest that was in Western Lake Erie. And then on the right-hand side, it now highlights the USGS project that corresponds with that point on the map. Another thing that when you're in this mapper, in addition to, let's say you're doing work in Lake Erie and you want to make sure that it's represented here because you don't see your project in the list, there's a specific form for each project type. So if you click on this plus research project in the bottom, it will bring you to it'll bring up a form on the right-hand side, and there you can enter your project title, your research topic, and you scroll down, you can give project information, contact information, and this mapper also has functionality of project links, images, linking papers, relevant, for example, relevant publication. It has the capacity to hold all of that project information, and you would submit that using this form. Another feature that's very, very helpful in this map is if when you're in the full project list, you can see in the upper right corner, there's this funnel icon, and that is a filter. You click on that, and it has specific filtering topics depending on if you're in the research projects list or the applied control project list. So for example, because this is the area that I study and have a strong interest in, I put up the filters and I see, well, who's doing biology, biology, life history areas of research for Dreissenids. I kept control method as all because that I wasn't looking to filter by that. And then I want to know what's going on in Lake Michigan. When I do that and I hit apply, it produced this list of projects and then these points on the map that you can see here. I clicked on one. I'm like, well, that sounds familiar because that's the project I submitted. And I click on that map and I can see that it is then highlighted on the project list on the right-hand side. And then if you click on details, it'll open up in that panel details specific to that project. In this one, I can see that, I'd love to put a picture up there. We put a logo of the organization as a default for all the projects, but I could instead have a cool mussel picture up there instead of the GLURL logo. I could add relevant linked databases or pictures, and all of those, if you scroll down here under research project details, you'd be able to get that information on any project you pull up. So along the lines of updating projects or giving feedback on the website, I'll point you to this box in the bottom. And it brings you to a form that has two purposes. One, let's say you can't find a function or you're trying something and it's not working. You would click on the IMC Research Coordination Mapper feedback. So you click on that lower box that's on the right. But I will spend more time focusing on the project update box. So let's say you are taking a tour of this and you find a project, one of your projects, and some information has changed or you want to add links, any of that, you can use this form to correct the information, such as contact information if something's changed in a project. You can add images, links to websites or articles, or upload publications. I put an asterisk here because we'd like to upload publications that you have permissions to share and that are open access and you're able to share them. So with that, that's kind of a quick whirlwind tour of this very interactive, but therefore it gets complex tool. So I hope you can take some time to play around in it and find the many features. Hope you find them useful for you to maybe find someone new who's doing research in this area, or to learn about what tools are actually being used or have been practiced in the field as an applied project. So now I'm going to switch gears to our other research group product. And that was to work with managers and researchers to rank Dreissenid mussel research priorities. The results of these we want to use to inform future research efforts. So to start out this process, we first developed a list of 12 priorities. This was a very iterative process. We identified common themes within the 2018 IMC strategy that Sam introduced in the beginning. We incorporated feedback from research group members, we refined the priorities, and we landed on 12 priorities in these broad categories of ecosystem impacts, control method development, and targeted control. We then developed a questionnaire for people to rank these priorities. So who responded? Well, we made a point we did this by invitation to reach out to gather people that represented a broad agencies, organizations, academic institutions, NGOs. So all the categories here are mentioned of the 94 people who did respond. We had a response rate of about 50%, which is fairly good for those of you who have put out surveys or questionnaires. And then as a bonus, we had extra people who were recommended or volunteered or delegated to do the survey. And as long as we felt someone had the qualifications to give good feedback, we included those responses as well. And about half were in the Great Lakes region and half were not, which is going to give us an interesting opportunity to compare between the two. And that's what I'm going to start doing on the next slide. So in the first round of the questionnaire, we asked respondents to rank the priorities within each of the three categories. We wanted to help people get to know the different categories first and the priorities within those. So they were asked to identify their first and second priority within those four. We took those weighted scores and summed them, and this is what we came up with. So for ecosystem impacts, we have the four different priorities within that group, and the highest ranked were how zebra and quagga mussels alter food webs and nutrients, and separately how they alter the environment. Now for all of these, I'm going to use a similar format for each of the three categories. And so first I'll give the broad results, but then we did break these out into Great Lakes or outside of Great Lakes. So in this case, you can see there are different responses that between these two within and outside of the Great Lakes. And it largely comes into these first categories, where you could see that a big difference is there was more outside of the Great Lakes. There's more importance on how the environment is affecting zebra and quagga mussels versus what the impacts are of zebra and quagga mussels. So there's definitely a shift in the weightings of those categories. Thinking on this, I think it could reflect that there's out west, it's an active invasion front, and therefore a strong interest in what environmental factors could be used to predict where dreissenids may establish. So I think it may have captured that different priority we have of a group with an established invasion versus one on the invasion front. For control method development, very strong priority that came out on top was developing selective control methods. And then the lowest priority is identifying post-treatment actions. When we broke this out into Great Lakes and outside of Great Lakes, there was good agreement and the general ranking of the research priorities in both groups, with selective control methods being the top. Now the final group of targeted control top priorities, or what would be a motivation for doing a targeted control in a region. Protecting fish habitat came out as the strongest, followed by protecting native mussels, and that's overall. However, when we broke this out into Great Lakes versus outside of the Great Lakes, the big difference here was in the infrastructure area. And a reason for this, thinking about it some more, is that in the Great Lakes, drysenid mitigation efforts are already built into water intake operations. And whereas out west, new invasions means new infrastructure needing to manage drysenid impacts, and that could be seen as a greater need and a greater economic hit. Then for the second part of the questionnaire, and to wrap up presenting our ranking priorities exercise, we then told the respondents, please rank all 12 of these priorities outside of the categories. Just you can rank them however you'd like. We had the top tier of importance, asking what's 1, 2, 3, 4. But then the next group of 4 is just you identify as middle tier, and then the lowest 4 are just lowest tier importance. And each of these rankings got a certain weight, a certain score. We summed up those scores, and this is what we found. Overall, what came out on top: developing selective control methods, how zebra and quagga mussels alter nutrients in food webs, and also protecting fish habitat. There are some other ones that are close behind there, but those are the top 3 that came out. And on the bottom, the lowest was reducing nuisance benthic algae as a reason to do targeted control, identifying post-treatment actions, and also minimizing impacts on infrastructure were among the lowest. So just looking forward, as I wrap up here, the research group, our next steps: compare the research priorities to our current research inventory, especially now that we have everything organized into this the project coordination mapper. And I'll from that identify gaps, say there are areas where we're not seeing any research on the forefront of these priority areas. And then ultimately develop a multi-year research agenda to address high priority research needs. So I understand we will finish there, and we will hold questions until the end for the panel. So thank you for your attention.
Thanks, Ashley. That was a great summary of all of the work that the research work group has been doing over the past 5 or so years. So wonderful. Yeah, and please enter any questions you have for Ashley regarding what she's presented into the chat, and we'll make sure to get to them after all speakers have presented. So it is now my pleasure to introduce our second speaker, Diane Waller. And while I'm doing that, Diane, feel free to pull up your slides. Dr. Diane Waller is a research fishery biologist at the U.S. Geological Survey Upper Midwest Environmental Science Center. Her primary area of research is development of control tools for invasive mussels and evaluating their effects on native and non-target species. Diane has been involved in the IMC since its formation and has seen how the Invasive Mussel Collaborative has both increased awareness of dry-seeded mussels and increased our communication and collaboration efforts among researchers and management agencies. Diane serves as the chair for the toxicity testing work group. And Diane, I see your slides. They look great. You can go ahead and take it away. Thanks so much.
Thank you very much for the introduction, Sam. I'm going to keep my video off just because we've had some internet issues here today. So with that, I'll get started. So this work group was formed in 2019, as Sam had mentioned, and the members that you see here shown on the left represent universities, state, and federal agencies from both the US and Canada. So in addition, the list on the right here is researchers that we called on to review proposed recommendations that the work group developed. So these outside reviewers have expertise in dry-sided mussel biology and aquatic toxicity testing or both, and also come from the US, Canada, as well as Europe, and represent private, university, and government agencies. And I just want to give a huge thanks to both groups for their time and efforts for this project. So as a major goal of the collaborative is to promote development of control tools for dry-sided mussels, it's really important to understand how control tools are being tested and evaluated. So this work group undertook a review focused on toxicant testing to develop a more standard framework that would advance consistency in methods and allow us really to be able to compare results across laboratories and among studies. We had 2 separate reviews, one for laboratory-based studies and a second for open water control projects. The work group tasks included reviewing how the data are being generated and reported across studies, identifying strengths and weaknesses of different approaches, and developing a draft of best practices. Also looking at areas that really need further attention. An outcome of the literature reviews and searches has been a database of studies that are now going to be housed in one place and can serve as a resource for researchers and also for management and regulatory agencies. So starting with the review of laboratory-based studies, these are the questions we had in mind in conducting the review and developing the guidelines on recommendations. So first, have standard protocols been used in dry-sided testing? What methods can be recommended that are specific for dry-sidids, and what areas of testing and reporting need further attention? So our process started with reviewing existing standard methods for other aquatic invertebrates and fishes. So there are ASTM guidelines, for instance, for conducting toxicity tests with aquatic invertebrates and native mussels. We looked at those guidelines that EPA has developed, OEC, Environment Canada, and CTAC. We then searched for published literature, also reports and gray literature of laboratory-based toxicity studies, then extracted data on all different aspects of how the studies were conducted, all the way everything from methods for field collection to the final assessment and how the results were reported. We then drafted recommendations on each aspect of testing and reporting, and then sent those recommendations to an outside panel of experts for their input. So the full database contains over 100 studies. We have since filtered out some of those studies whose objectives were more focused on bioaccumulation or effects of contaminants on dry synods. Of those that remain, we had 74 studies that were really focused on evaluating toxicants for their effects on dry synods, really related more to control research. And here's how the studies broke down by life stage. You can see the majority used post-settled mussels. This means adult and juvenile mussels. Some tested the range of life stages, so the total here is greater than 100. Just looking at this percent of papers though, one thing that became clear very early is that we do need additional work on those pre-settled or early life stages. As for standard methods, the percentage of studies that cited a standard protocol was just a little more than 30%. Now, it doesn't mean that the studies weren't valid and weren't following some standard guidelines. It just means that 66% of them did not cite what standard methods might have been used. So following the review, we summarized the data and looked at it compared to what these standard toxicity guidelines are. And from that summary, we identified testing parameters that are specific to dry synods and really varied across studies. So these topical areas are shown here, everything from how to confirm that you have a zebra mussel versus a quagga mussel, down to, what are the specific test methods and what are the criteria for mortality. So for each of these areas, we developed suggested recommendations based on literature, reports, standard methods, as well as conversations with researchers and the experience of people in the work group. So here's an example of a key area that varies among studies and for which there really is no clear best practice. And that is how to determine that test organisms are in good condition for testing. Mussels might be collected at different times of the year. They might be held in a lab for different lengths of time before they're tested. All of these can affect what their condition is during the test and how they might respond to the toxicant. So we might over or estimate what is a lethal dose of a molluscicide based on what the mussel condition is at the time of testing. So you can see on the table here that about 28% of studies didn't report how the suitability for testing was determined. Of those that did, the two main criteria were attachment to a substrate or what the mortality was during holding. A couple of papers included observations of normal behavior like filtering and their response to probing. So based on the criteria that had been reported, the work group developed recommendations for this criteria. So this is shown on the right. This is what our review panel was asked to basically comment on and select which of these recommendations or criteria they would recommend. We also offered a space here for them to give additional comment or feedback. So here's an example of one recommendation that we drafted for this component, suitability for testing. So it is a combination of standard methods, the review of these published studies, and our input from outside reviewers. So the first three bullets that you see here come from ASTM guidelines. Bring mussels into the lab and hold before testing, monitor them for normal activity, and also recording what the mortality is during that holding period. These last two bullets were added as Dreissenid specific criteria, and these were most commonly cited in studies and those that were recommended by our expert panel. So this includes resistance to valve opening. This ensures the mussel isn't weak or the valves in fact aren't empty. So for attachment, the mysis is severed and mussels are placed in a holding tank overnight. If they don't reattach, they're considered unsuitable for testing. So our recommendation is to also include at least one or both of these last two criteria. We have a full set of guidelines and recommendations that will cover all aspects of testing. As I showed on the previous list, this is everything from collection methods, verifying species identification, holding and acclimation conditions, what the test methods are and response measures. And really importantly, what we found is the information that's being reported is perhaps our largest weakness here in conducting studies and reporting them is what parameters we also need to include during the, to measure during the testing. So this document is going to be sent out later this month for final review by our expert panel and will be made available in a publication as well as on the website. So the second review here was the meta-analysis of open water Dreissenid control projects. And this was led by Angelique Dahlberg as part of her doctoral research at the University of Minnesota. The review is looking at any and all treatments for Dreissenids that occur in open water and involve the use of a molluscicide or pesticide. So it does not include things like drawdowns, manual removal events, or anything in closed or industrial setting. The goal here is to search for broader themes from these control projects and really shed some light on how different elements can affect long-term success. So for this work, Angelique collected information on all projects in North America. She broke them down by project phase and project goals. So that included looking at what work was done pre-treatment, the actual methods for treatment, and what was measured and reported and post-treatment. So here's a table that shows just some of the information. This is 33 treatments that have been applied in 23 lakes since 2006. And the projects were broken down into, by project goal, into 4 categories of rapid response eradication, an established population eradication, a suppression, or a research project. So a review of really lessons learned in these open water treatments is currently under review by the co-authors. And this is going to be a really valuable resource for guiding future open water control efforts. So for example, one finding was that successful eradication in a partial lake treatment really needs to include pre and post-treatment survey methods in identifying all life stages that can be present within the water body. And those surveys have to have adequate coverage across the water body to really hone in on the size of area that should be treated. Another missing element in many of these projects was collection of non-target and native community response data. And this is really important information for decision-making on what are the pros and cons of the treatment that a regulatory agency might need to consider before permitting an open water treatment. So this is just an example of some of the kind of lessons learned that are identified from this review. So there will be a brand new page on the IMC website that will house the products of this work group, both the laboratory and open water reviews. So this is the page shown here, and it is also found under the research and projects drop-down window. It will include 2 tables, one for the laboratory-based studies and one for the open water treatments. The complete metadata will also be available in an Excel spreadsheet that can be downloaded in the highlighted blue here tab. And right now what we have available is the laboratory-based toxicity trials. The plan is to keep this database updated annually in order to keep it current. So here's a portion of the laboratory-based table that's developed. First of all, there's an active link to each of the papers. The searchable columns include the authors, the date, life stage tested, the control agent, the response measure, non-target species, and the journal. So for instance, if you're looking for a study with a specific toxicant or molluscicide, you can enter it in the search bar. I entered Zequinox here, and you can see some of the studies that came up in which Zequinox was evaluated. Or if you're interested in non-target species that have been tested along with dreissenids, you can enter the species name or general group name. So I entered walleye, but you could also more generally look for fish or mollusks. So you can see shown here some of the studies that have included testing of walleye along with dreissenids and toxicity testing. So we hope that researchers as well as regulatory staff and resource managers will find it useful to have these studies in one place and be able to search for specific keywords related to a study. So again, the products from the work group will be available on this new webpage at the address shown here at the bottom of the screen. So you'll see it also is found under research and projects. The review of open water treatments with a summary of what's worked and areas for improvement will be added to this as well as the table of open water treatments. So I just want to mention you can send questions to or inquiries for more information to me at the email shown here or to staff at GLC. And I also just want to give a great big thank you to GLC staff for their awesome work to help coordinate this work group and develop the products that we're now rolling out. And also to our staff here at UMass for the hours of pouring through papers and extracting the details. And I will also leave questions to the end. So with that, thank you, and I'll see if I can turn off here.
Thanks so much, Diane. And just to let everyone know, the page that Diane was just mentioning is now open to the public. So I put the link in the chat if anyone wants to check it out. And with that, I will introduce our third presenter, Jeff Tyson. And Jeff, you can go ahead and pull up your slides while I do so. So Jeff is a program manager for the Great Lakes Fishery Commission in the Fisheries Management Director and the Fisheries Management Directorate. Jeff actively works with the fisheries management agencies through the Lake Committee and Lake Technical Committee structure to facilitate cooperative fishery management across the Great Lakes Basin. And Jeff serves as the chair for the Coastal Site Priorities Work Group. And okay, Jeff, it looks good. I can see your slides and you can take it away. Thanks so much.
Okay, great. Thanks, Sam. And thanks everyone for hanging around for an update from the last work group here. I'll be presenting information on the Coastal Site Priorities Work Group progress and products on behalf of the Coastal Site Priorities Work Group, as well as my co-authors. And as you heard before, the Coastal Site Priorities Work Group, along with the research and toxicity testing work groups, were all stood up in the winter of 2019-2020. The membership of the Coastal Site Priorities Work Group is very broad, comprised of federal, state, tribal, and provincial folks, as well as others. And these folks have done a fabulous job of kind of helping guide the work group itself to development of the products that I'll be presenting. And you can see the Coastal Site Priorities Work Group right here along with the research and toxicity testing work groups. And this is an actual copy of the charge from the IMC steering committee to the Coastal Site Priorities Work Group. Essentially, the group was to review information on where dracaenids were impacting Great Lakes resources, identify criteria for evaluating sites based on this information and other considerations, and develop and implement a system for prioritizing sites for potential management activities. And initially, the work group had some discussions about what the products would look like. And at the end of the day, the products that we'll be providing to address this charge will be a prioritization and screening tool that can be used based upon management research agency specific needs. So be flexible enough to account for any additional needs that other management agencies might have moving forward. One of the first steps that we had to do was to inform or to decide what platform we would use, the scope and the resolution of the prioritization. And fortunately, we had a geospatial platform out there that was already available, and we had some expertise in it with the Great Lakes Aquatic Habitat Framework. The Great Lakes Aquatic Habitat Framework is a series of hierarchical nested grids that range from 30 meter up to 9,000 meter resolution. You can see on the right-hand side of the screen the classifications associated with GLAF, with the nested grids, the 30 meter nested within the 1,800 meter nested within the 9,000 meter. Additionally, there are a number of zones already defined, habitat zones defined within GLAF, which we utilized for the prioritization process. The scope was determined through the charge. It was basin-wide, but the work group did constrain that to coastal, terrestrial, coastal margin, and nearshore areas, largely in recognition of operational constraints associated with any potential management actions that might occur down the road, plus to make it a little easier to handle a fairly large data set. The resolution that we settled on was initially the 30 meter resolution, but we did agglomerate that 30 meter resolution out to a base layer of 90 meters, and then also utilize the 1,800 meter and 9,000 meter resolutions for the prioritization process itself. The next step in addressing the charge was to review information on dracaenid impacts and establish criteria. And the work group really discussed how we would review that information and essentially settled on a determination that dracaenids were largely ubiquitous across the basin except for Superior, and that the impacts would largely be associated with the distribution of the primary criteria that we selected or the things that we cared about. So we utilized the Invasive Mussel Collaborative strategy to advance management of invasive zebra and quagga mussels to identify those primary criteria. And we pulled 5 criteria out of that strategy that were repeatedly mentioned within the strategy. And it's really consistent with some of the priorities that were identified by Ashley through the research prioritization process as well. But important fish spawning and nursery habitat impacts were identified as important, unionid impacts, Cladophora distribution and impacts, and then threatened and endangered fish species impacts, as well as water intake infrastructure. The work group also identified a number of secondary criteria for consideration, particularly from an implementation standpoint. However, a number of these secondary criteria weren't available at the basin-wide scale. So we included that within the documentation for consideration as folks pick up this screening tool and start to consider potential management actions. There's a number of additional secondary criteria that will need to be considered within the context of any management action. So that's there, but it's not mapped. So next we'll move on to the primary criteria. The first primary criteria that we mapped across the basin was important fish spawning and nursery habitat. And we utilized various sources from the literature, including a fairly recently published synthesis from Ebner et al. We also utilized information from Goodyear et al., Zhu et al., Roseman, as well as other sources. These important fish spawning and nursery habitats really focused on specific species like lake whitefish, cisco, lake trout, and walleye. And the way we handled it was we assigned essentially a standardized range to each of those 90 meter grids across the basin, with 0 indicating important fish spawning and nursery habitat was absent, where 1 was indicated presence of important fish spawning and nursery habitat. And so it was essentially a binary value. But I do want to note that the list of important fish spawning and nursery habitats aren't exhaustive, and these can be updated and edited moving forward, but they are really the most important that have been identified through a number of literature sources, as well as best professional opinion, and they are basin-wide. And this map on the right-hand side shows an example from Lake Michigan, where you can see some of these important fish spawning and nursery habitats. It's identified in a number of those sources, with the red areas indicating that they are fall within that important fish spawning and nursery habitat. The second primary criteria that we developed was a distribution of unionid refugia across the basin. And because there's not a basin-wide unionid assessment program, we estimated the distribution of unionids through an ecological niche model, MaxEnt. We used a number of environmental layers from GLAF itself to predict where potential unionid mussel refugia might exist. Depth and fetch were the 2 primary environmental layers that we utilized. We did look at a number of others as well. And we did use training data from Lake St. Clair, Erie, and Ontario, and that was presence only data. And this was provided by John Bossenbrock. And it really, the method that we used here was very consistent with the method that Bossenbrock used and published in 2018. We just extended it basin-wide. Again, we standardized the distribution of unionid refugia to range from 0 indicating absence to 1 indicating presence, and we use characteristics of the information to determine the cutoff for suitable versus unsuitable unionid refugia habitat. The third criteria that we developed was Cladophora distribution across the basin, and thankfully we really didn't have to develop that. Michigan Tech Research Institute and Brooks et al. Had already developed a distribution map of Cladophora across the basin, and it's based on a depth invariant algorithm using Landsat. We utilized information from 2017 to 2020, and really the maps indicate the extent of submerged aquatic vegetation and optically shallow areas. So there were some areas that we didn't get a good estimate of SAV, i.e. high turbidity areas. But in general, the distribution maps indicate the distribution of Cladophora across the basin. And the information was essentially categorized into not present, light coverage of SAV versus dense coverage of SAV. So we standardized, again, that to range from 0 indicating absent to a 1 indicating dense, with a categorical value of 0.5 for light Cladophora coverage. And this map again shows that distribution for Lake Michigan. We did this for all lake basins. It's just easier to see for individual lake basins. The fourth primary criteria that we developed was one for threatened and endangered fish species. And we canvassed all state and provincial agencies for listings by counties. And then we screened the species that were listed for each individual county, and selected only those species that likely would be inhabiting the Great Lakes proper. And then we developed a county index that was generated and then standardized to range from 0 to 1. The index essentially was developed at the county level and was based on the frequency of listed species, so threatened plus endangered, plus the frequency of endangered species. So it was more heavily weighted towards individual counties that had higher frequency of endangered species listings. And again, that was standardized to a continuous value to range from 0 to 1. And you can see some of the darker red areas here have a higher threatened and endangered species index value, as opposed to some of the lighter pink areas across the basin. And then the fifth primary criteria that we developed was water intake infrastructure. And this information was compiled from state, provincial, and federal data sources. We essentially took the point estimates for each of those water intake infrastructures water intake structures, I guess, and applied a 1 kilometer buffer around each of those point locations. And again, developed a binary value for this standardized to range from 0 to 1, with 0 indicating absence and 1 indicating presence. And it's kind of difficult to see on this map because there's really only one, at least in the zoomed in area, one important water intake infrastructure area right here in Grand Traverse Bay. For the prioritization process, we essentially summed the 90-meter individual criteria scores across each of those individual primary criteria. So it was the sum of important fish spawning and nursery habitat, unionid refugia values for each of those 90-meter score, 90-meter grids, the Cladophora distribution, water intake infrastructure, and threatened and endangered species. And what this really indicates is locations where these multiple criteria spatially overlap, and would indicate locations where if Dracaenads were present, they would be having likely an impact on some of the things that we care about. Some of the challenges that we had with the development of the prioritization process included the fact that we were dealing with 90-meter grid sizes. So the data file size was very large, 14 million records that were pretty unwieldy and difficult to use for any additional post-processing. And then some of the criteria, or at least the prior 90-meter prioritization values, were relatively widely distributed. There was some evidence of clumping, but they were widely distributed. And also we had to recognize some of the interannual variability associated with some of the primary criteria like Cladophora, which likely vary spatially on an annual basis. So the solutions that the work group came up with to address some of these challenges, including scaling from that 90-meter up to the 9,000-meter resolution, but also retaining the 90-meter, or at least an index of the 90-meter grids to indicate which criteria were influencing the overall 9,000-meter priority scores. And the way we scaled up was essentially we took the maximum 90-meter coastal site priorities index grid value and applied that to the 1,800-meter grid that it was nested within. And then we conducted some additional analyses on those values, and really any value that was greater than or equal to 1.7 indicated relatively high overlap with those primary criteria. And so we essentially counted the frequency of the 1,800-meter grids that were nested within each 9,000-meter grid that had values greater than 1.7. Then we used Jenks natural break by lake with 5 classes at that 9,000-meter resolution to identify moderate and high priority sites for consideration for implementation of management actions based on the primary criteria that we had. We also did include a primary criteria index or indicator value in the attribute table that would give you a sense of essentially which 90-meter criteria values were contributing most to that 9,000-meter prioritization score. And at the end of the day, this is what the prioritization looks like. You can see moderate to high priority grids based on those primary criteria across the Great Lakes Basin, ranging from Superior down to Ontario. We've got a concentration of high priority grids within Northern Lake Michigan, Northern Lake Huron, Georgian Bay, Western Basin of Lake Erie, and also the Eastern Basin of Lake Ontario. The prioritization process that we developed is essentially intended as a potential guide for future work, but is not intended to constrain work that has or will occur in the future or other locations based upon different criteria or these criteria that are applied differently. So hopefully we created the prioritization process with enough flexibility that folks could take advantage of additional information if they chose to, or it could be updated in the future as well with new information. And then lastly, I did want to show you what the attribute tables for each of the 9,000 meter grids look like. Recall that I mentioned we included an indicator for each of the primary criteria at that 90 meter resolution that would indicate the influence of each of those on the overall prioritization score at the 9,000 meter. And you can see a range of values here indicating, you know, some of these grids are influenced fairly heavily by important fish spawning and nursery habitat. Others not so much, but mostly by unionids as well as threatened and endangered species. So it gives you a sense of who's impacting what at the 90 meter level within that 9,000 meter prioritization process. All of these products, including the documentation, the metadata, as well as the individual primary criteria layers and the prioritization layers, will be available in the future on the Invasive Mussel Collaborative website. We have a call tomorrow to start talking about the process for getting this information out more broadly and making it more available to folks. But that is not done yet, so we're behind some of the work that the research work group and toxicity testing work group have already posted out there. Lastly, I'd like to thank the Coastal Site Priorities Work Group for all their work and heavy lift on this, the Invasive Mussel Collaborative Steering Committee Corps, and also the Great Lakes Commission for all their great support, including Sam and Jill and Ceci and Erica. Michigan Tech Research Institute for providing the Cladophora coverages, and I'd like to encourage you to visit the Invasive Mussel Collaborative website. At some point, we'll have some products posted up there that folks can take advantage of. And with that, I will turn it back over to Sam and try to stop share. Oh, somebody stop share for me. Very good.
Yep, I took control, Jeff. Thanks so much.
Okay. All right, thank you. And I guess we'll open it up to questions or comments.
Yeah, thank you to all of our presenters today, Ashley, Diane, Jeff. That was really great. Thanks for taking the time to present all of the products that the various work groups have been striving towards. And I really just want to give a special acknowledgement to all three of you today. Ashley, Jeff, and Diane have served as work group chairs, as I mentioned, but this is on top of their positions with their various agencies, and their leadership, along with the dedication of all of the work group members who are participating, has really resulted in the advancement of our Invasive Mussel Collaborative strategy through the products that they've presented on today. So we will move on to questions now. So any questions that you have for our presenters, you can go ahead and type into the chat box and we will be glad to answer them. And I guess while we're waiting for folks to type in their questions, I do have one question, Jeff, for you. How easy is it, or what kind of is the process do you foresee going to be for folks who are looking to either play around with the tool, or if they'd like to add additional variables or anything like that? What's the process, and how easy is that going to be to do?
You know, the process for adding, particularly if you're working, if you're not working at the basin-wide scale, the process for adding additional criteria should be fairly easy and straightforward, particularly with the Great Lakes Aquatic Habitat Framework and the nested series of grids. So in essence, if you've got a shape file, a coverage shape file of some fashion, a polygon shape file that you can place into the geospatial framework, it's really easy to assign essentially that additional data to that 90-meter grid. So it shouldn't be too challenging. It will take, you know, some GIS expertise, but it's not so challenging that I couldn't do it. So
okay, that's great.
And I think there really will be an opportunity to even update the primary criteria that we've developed to date with additional information as that comes available. So I've been talking with some of the folks at the Nature Conservancy about some of the work that they've been doing modeling lake whitefish and cisco spawning habitat across the basin. And I think that'll provide some additional information to help inform that important spawning and nursery habitat layer.
Thanks, Jeff.
Mhm.
And Diane, I have a question for you. What is going to be kind of the process or timeline for keeping the database up to date as new research comes out, or as there's new field tests or field studies, open water studies? What's that process going to look like, do you think?
Yeah, so I think it would be an annual update that, well, I think you guys do a great job of updating publications as they become available, and it will really just be more of a targeted effort to look for those that are, relevant for either the laboratory-based toxicity studies and also the open water control projects. Now, those I think will take more of a concerted effort because many, sometimes they aren't necessarily in a published report. I know Angelique spent many hours on phone calls and just interviews to dig out some of that specific information on an open water treatment. So that we will hopefully become aware of also as people maybe are adding them to the mapper. So there should be some cross-referencing there between the mapper and what we have in the database files.
Yes, it's all interconnected, absolutely. And then a question kind of on the research work groups. I'm not sure who wants to take it, Diane or Ashley, but in terms of the prioritization survey that was done, how is that information going to be available? Because I can imagine there's going to be some interest from managers and policymakers across the region on what's a priority, both within the Great Lakes, but also across North America.
Yeah, I'll take first stab at that. And that's in our next steps to develop that targeted multi-year research agenda, that the top priorities would be there. I also think, though, that just to have full transparency and to have the the full listing of all 12 priorities ranked, I think that we could put together a one-pager and have that available on the website more immediately. That's something that we just hadn't polished up yet to have as an outward-facing product, but that would be easy to put together if it sounds like there's interest.
Thanks, Ashley. Yeah, I think there is. Any other questions? Go ahead and you can send them directly to me, as some of you are doing, or you can go ahead and put it into the general chat. Okay, and Jeff, one more question regarding the different layers. Which I guess I'm forgetting what they are. The 9,000. Which one do you foresee being most applicable to those looking to identify sites for potential management activities, and kind of what are the pros and cons? I think is what they're getting out of deciding between them.
Yeah, I think, the 9,000 meter resolution probably provides you with the best sort of regional guidance towards site selection. I think the 90 meter will be useful, but I would presume that if you are looking at specific or individual locations, there's going to have to be some additional ground truth work that occurs. So you can utilize the 9,000 meter to get a pretty good sense of where you've got significant overlap of these individual criteria. The 90 meter will be very helpful, but it's still not going to preclude the need for additional site-specific reconnaissance work to sort of verify what the individual layers are saying. So, but I think it's a combination of both. The 1800 meter is essentially a scalar. It's moving things up. So there's not a whole lot of information in that layer, but it's just the way that we scale things up.
Thanks, Jeff. Okay, Ashley, I have a question for you. Wondering what your thoughts were on why post-treatment actions were ranked as one of the items of least concern for both the Great Lakes region and out west.
Yeah, it's interesting to bring that up because in putting this presentation together, I was thinking of the same thing. You know, I see it as, I don't know, maybe it's maybe something that we take for granted and that it's important and that's part of bigger projects, and that when it was called out as a separate action, people felt that, well, the broader projects and those other efforts were higher priority, but maybe thinking we should be doing that post-treatment work after anyway. It could be maybe not fully understanding what that could include for the post-treatment actions. You know, for example, that could include if you're reducing mussels and you need to promote native mussels, any actions to do to protect other aspects of the habitat to promote native mussels and also reseed native mussels. And maybe that wasn't, those kind of actions weren't implicit in describing the priorities. So I think it's one that in revisiting it, we didn't have enough description to it, or people thought it was maybe already wrapped up and would be part of other efforts.
Thanks, Ashley. Okay, Diane, here's one for you. When reviewing the metadata, is there any one treatment which seems to have the best potential for use in treatment of zebra mussel infestation of freshwater lakes?
Yes, I was just thinking about that response because one of the, and I'm assuming this is in reference to the review of open water treatments. So one of the findings is that really all of the products that have been used can effectively kill mussels. It's really more about, you know, delineating the area for treatment. If you want eradication, to know what area you need to apply to so that you're not missing, for instance, villagers or smaller clusters of adults outside that treatment area. Now, most of the open water work has been done with copper-based products, particularly EarthTech QZ is registered for open water use, so there's just more information on use of it. So as far as a chemical-based molluscicide right now, that is probably the one that is most often being used. I'm not advocating for any product one over the other. That's not what we're doing here. I'm just saying that one has the most information available on its use in open water treatments. But again, I would say that like Zequinox is also an effective product. It's more about the pre-treatment and follow-up monitoring that really can lead to whether or not the project itself is successful.
And I'm sure, Diane, once we are able to publish all of the summaries and information, that's going to be able to help people answer a little bit more of those questions on their own too.
Yes, exactly. Yeah, we have different definitions of a successful treatment, so it will really depend on what your management objectives are with it.
Okay, we have another question, and in this one, it seems like it could be answered by any one of our speakers. So kind of an in-between question. The databases look great. I'm mostly interested in methods to protect piping and infrastructure for water intakes in affected watersheds. Where would some of the data on who is using what technology to control zebra and quagga mussels in affected watersheds and its efficacy? So where could they find that data? And it seems like that is between some of the databases.
So is that a question on use on infrastructure treatments? Did I hear that correctly?
Yeah, they're most likely interested in methods to protect piping and infrastructure. So it looks like controlling in areas where that's a significant threat.
Yeah, at this point, our databases don't specifically include closed systems, but I think the mapper, you could go to the mapper and see where some of those control efforts are occurring for infrastructure, and that could lead then to getting more of that information. And the Bureau of Reclamation is a great place to start.
Yeah, thanks for that, Diane. Looking through the mapper as far as filtering it, there isn't an option. The water body focus areas are on natural water bodies in the lakes or the Great Lakes specifically, or rivers and streams. We don't have a infrastructure or kind of the internal waterway option to search by. You thinking about projects? I don't know if we've had that many that spoke to that specifically. It would be more pointing to some of the technologies that are being used by infrastructure. But yeah, that's something, if you want to follow up with me, I can help maybe look through if our filters aren't getting to those, we could help connect you. And I think Diane was right on of mentioning Bureau of Rec would have the best sense of efforts there.
And maybe this is another tab we can add in the future to our database of an additional third tab after open water treatments.
Yeah, I absolutely would open to having that as a category of searching where these efforts are taking place. And that's exactly the kind of feedback that you can use on that, the project or the feedback form that's on the website. If there's a category functionality that you think could be used, even if it's something that would take us a little bit to develop, it's worth discussion, and please bring it to our attention so we can discuss it as the research group and keep evolving this tool to meet everyone's needs.
Thanks, Ashley. And they said, and thanks, love the idea of a third category in response. I am not seeing any other questions, so I think we can probably wrap it up. I just want to thank our three speakers once again. Their emails are up on the screen, so if you do have any lingering questions, feel free to reach out, or you can reach out to us at the commission, who support and provide the administrative support to the Invasive Species Collaborative, or to the Invasive Mussel Collaborative at mussels@glc.org. And before we wrap up, I do just want to say there's opportunities to get involved. You can attend additional webinars. You can go through and look at our tools, submit data. We have lots of opportunities for involvement, both from the perspective of the interested public, but also we're really looking to engage researchers, managers, people who are on the ground doing the work. So these are the tools that we have produced for you. Okay, and so thanks again to all the participants who are watching this webinar, either today or in the future, as this webinar will be recorded and available on the Invasive Mussel Collaborative website within the next few days. And if you have any follow-up questions or comments, you can feel free to reach us. Again, our email at the Great Lakes Commission is mussels@glc.org. Thanks so much, everyone. And thanks, Jeff, Ashley, and Diane. Appreciate it.
Yeah, thank you.
Yep. Thanks all.
Bye, everyone.